I'm going to be talking about how NASA monitors the ocean from space and from ships, and how you can do it too. I have some tools at the end to share with you. Okay, so this is the biosphere. Other people have showed it in their talks, but basically you see plant life on land and in the ocean. Plant life on land, it's very intuitive, it's green for more productivity. And in the ocean, we have higher productivity shown in the lighter blues and then the yellows and the reds, whereas the ecological deserts, or the very low productivity, is shown in purple in those central gyres of the ocean. NASA has been continuously monitoring ocean life from space for the past 20 years. We just had the 20th anniversary of the satellite that started doing that. And from this, it's really revolutionized our understanding of what drives life in the ocean. And you can see, these areas off of river plumes where nutrients are flowing in from the Amazon, for example, or where sand from the Sahara is blowing out over the ocean and fertilizing, or areas of upwelling, or where you have islands and you get sort of topographic upwelling. And we've observed seasonal cycles that you can see here. It's like the Earth is breathing. And then smaller scale features, mixing eddies. Hurricanes also stir up nutrients and lead to blooms. Next. And so this shows the ocean currents. And here these are shown, the colors represent the temperatures, and then you can see the motion. And this is what I do for my research. I look at how physics is impacting biology. So even though you can only see horizontal features on this visualization, what's happening is it's causing vertical motion that's bringing nutrients from depth up toward the surface in some of these features. So for example, where you have cooler water along the equator, that means upwelling has brought nutrients up to the surface, corresponding to the higher productivity you saw in the last visualization. Next slide. So the other way we look at, to match what we're seeing from satellite to what we see, to equate it to what's in the ocean, we have to take measurements at sea. And I was at sea last year, January and February, on the Schmidt Ocean Institute ship Falkor. We were in the Pacific. It was an expedition called Sea to Space, and we were looking at phytoplankton diversity. So we were taking optical measurements to compare to what the satellite was seeing, and then we were also taking measurements to look at the marine carbon cycle. So there's a lot of information on that site. Yeah, next, that's fine. So we went from Hawaii in January over toward the west coast and up. And these are images collected in a flow-through system. So this one was collected off Hawaii, and this one was collected off of California. This is from an instrument called Flow Cytobot. And so these are greatly magnified here, but basically these are phytoplankton. And just for reference, this black dot is a speck of dust that was on that microscope. And so you can see that the black dot is much more similar to the sizes off of Hawaii than off California, meaning we had much smaller phytoplankton off of Hawaii and much larger phytoplankton and something called diatoms forming chains. Those are phytoplankton that are, all phytoplankton form the base of the marine food web, but some phytoplankton are preferred more by fish, and so diatoms support a productive fisheries off California. Next slide. So the other thing we were doing at sea was to observe sinking particles. So this is these are sediment traps, and we were deploying them off the back of the ship. We had two different kinds of sediment traps, and they were being put in the water and then lowered to about 100 meters or about 300 feet. And they drifted for several days, and then particles from the upper ocean just sank into them. And then after 3 or 4 days, we would haul them up and see what settled in them. And this big block here had an iPhone camera in it that this scientist had, Melissa Oman from University of Rhode Island, she had worked with an engineer to put an old iPhone in the bottom of this block. And I'm going to show you the animation that she took in a minute. Next, please. And here's the animation from her iPhone. So you can see, we call this marine snow. This is the detritus and the phytoplankton and other things kind of raining down from the surface ocean, and it was falling into the sediment trap. But why this is important is that some phytoplankton, they uptake carbon dioxide or carbon, and then if they sink, they can remove that carbon, some of which came from the atmosphere, and they can remove it down into the ocean depths or into the sediment and keep it out of circulation for thousands of years to longer. And so we're really trying to understand the role of this. Each phytoplankton species behaves differently with respect to the carbon sink, and we're trying to understand that, and that's why we're doing studies like this. NASA has just funded a big field campaign called Exports that's going to start next year to specifically look at this issue of how much carbon is sinking and how can we then relate that to satellites. If we can tell what kind of phytoplankton are at the surface, can we then estimate through models and other observations, can we model what's sinking? Next. Oh, and so we were in an area of the Pacific that's apparently known as White Shark Cafe. So one of the instruments, when we pulled it up, it was the sediment trap. There were 4 sediment traps originally, and it came up and one was broken and the other 3 were missing. And one of the line handlers got a shark tooth in his finger, or in his glove. Thankfully, he was wearing a leather glove. And this is the piece of shark tooth. And it was later identified by an ichthyologist as being a white shark. So for some reason, the people who study sharks have tagged them and noticed that they hang out in that area in wintertime. They seem to be either breeding or something. And that's an area of active research. But we unwittingly sort of bumped into that area as our sensors were drifting. But the engineers on the ship were great. They rebuilt the instrument, and we were able to redeploy it after this. So there's a lot of challenges when you take measurements at sea. Next. So this is another little project I've been involved in. We take global satellite data and we create a visualization with a very short, so it's 2 minutes long, and it explains, what are phytoplankton, why should you care, how do we measure them from space, and we use data sets that you can use too. These are all online, and I'm about to show you how you can use them. But this, basically, it shows you the 20-year time series from a site called NASA Earth Observations. It explains, where the phytoplankton are more prevalent or less, and then what causes them to bloom. And then we draw the connection between satellite observations. Notice the low productivity areas in the circles, and then higher productivity along the equator and toward the poles where there's more nutrients when there's sunlight, greater mixing. And then this runs through showing you, how we take measurements from sea as well. And then we combine all of that into models. And so at the end, this is the instrument called the Conductivity Temperature Depth Profiler, and we take water samples, we look at them under microscopes, and we put it all together. And this is a model that's done by MIT and JPL. It's called the Darwin Model. And this is showing, in red are the diatoms, the larger phytoplankton that have sort of glass-like shells. And these are pictures of them under a scanning electron microscope. These are samples that we collected on that cruise in the Pacific. So you can see they're prevalent in areas where there's lots of nutrients. And then dinoflagellates, these are again from the cruise. They're more prevalent in the areas that are shown in blue, or yellow. And then these are the very tiny ones. Synechococcus and Prochlorococcus are called cyanobacteria. And they're very common in areas of low nutrients. And then coccolithophores are not shown by the model, but they're important because their shells are made of calcite, which is like chalk. And they're very sensitive to the changing chemistry in the ocean. So acidifying ocean we're studying what, in lab studies, they have not done well. Their shells become thinner. So that is the climate bits. And then next. And now I want to show you how you can look at this data too. This is a resource called NASA Earth Observations. And because we went on the cruise and I've been talking about phytoplankton, I'm going to show you an example of comparing chlorophyll and sea surface temperature. But it has there are over 50 satellite data sets in this tool. Here's the website. And you can take a very quick look. It has an online analysis tool. So I'm going to walk you through a very simple analysis here. And then you can go and try it. And I have some postcards that I can give you at the end if you're interested to try more examples. But let's say we're going to compare chlorophyll concentrations and temperature during that cruise we took. So if you go to the tab that says Life, you can click on chlorophyll concentrations. And under the tab that says Ocean, you can click on sea surface temperature. And some things are in multiple categories. I think chlorophyll is in both Ocean and Life. And then you pick the year that you're interested in, which is 2017, and I'm picking February for when we were out on that cruise. And then you add it to your analysis. You could even do this on your phone. And then you go to the chlorophyll data set, you pick the year, the month, you add it to the analysis, and you'll notice the number on this tab changes as you add to analysis, and you can compare up to 3 at a time. So it's a very simple tool. It's for taking a quick look. Then you launch your analysis, and you click Analyze, and you select your region. And so I'm picking this area where we did the cruise. You can either do it through a box with, picking your corners, or you can add your coordinates in, and then you launch your analysis. And there's a lot of options once you get your region of interest. You can click on Zoom or Data Probe, and here I'm showing a transect. So I'm drawing a line from Hawaii to the west coast where we went on our cruise, and you can see temperature was warmest in Hawaii and sea surface temperature decreased as we went toward California. Colder water from the upwelling. And then chlorophyll along that line went from fairly low, the value is about 0.09, and then it really spiked off of California in the productive waters. So this is it's a simple tool. It's a way to take a quick look. You could it's especially designed for non-scientists, but scientists could use it too. For example, when I was at sea and we had low bandwidth, I used it just to do a quick check of where we expected to get higher chlorophyll. Then you can do other analyses, like here we're selecting this region off the west coast and making scatter plots. And if you select an area, if you want to highlight a range of data, it'll light up in magenta. So if I want to look at where did I have high chlorophyll but cool temperatures, that's this area here. And then I could do it for high chlorophyll but low temperature, or warmer temperatures. And you see that's off Southern California. We have a poster tomorrow in the IN, the informatics area, number 40, and we'll be talking about these tools there. So, please come if you have any questions, or I think I have a little time. No, I don't have time for questions. Okay, and I have postcards, so if you're interested in this, there's more examples and links to the resources. Thank you very much.